The Justinian Plague, which first struck the Byzantine Empire in 541 AD, has long haunted historians and scientists alike, leaving a trail of devastation that reportedly claimed millions of lives across Europe, the Middle East, and North Africa. For centuries, the precise origin and biological mechanisms of this pandemic remained a mystery, often overshadowed by the later Black Death. Recent breakthroughs in ancient DNA analysis, however, are shedding light on the deadly bacterium Yersinia pestis, confirming its role in the Justinian Plague and offering unprecedented insights into how it spread and why it was so lethal. Researchers have recovered fragments of Yersinia pestis DNA from human remains buried in mass graves dating back to the 6th century, allowing scientists to reconstruct the pathogen's genetic blueprint. According to these findings, the strain responsible for the Justinian Plague possessed genetic features that made it highly virulent, yet somewhat different from the strains behind the Black Death in the 14th century. Experts suggest that environmental factors, such as widespread famine, war, and the dense urban populations of Constantinople and other cities, likely amplified the outbreak's severity. Interestingly, genomic evidence also hints at a series of mutations in the bacterium that may have enabled it to survive in multiple hosts, including rodents and fleas, facilitating rapid transmission across continents. While some researchers argue that these mutations alone cannot fully explain the scale of mortality, the combination of pathogen virulence, human vulnerability, and socio-political instability appears to have created a perfect storm for one of history's deadliest pandemics. These insights are not only important for understanding the past, but also provide valuable lessons for modern epidemiology, highlighting how environmental and societal conditions can influence the impact of infectious diseases. Some historians note that contemporary accounts describe sudden waves of fever, chills, and swollen lymph nodes consistent with bubonic plague, and the ancient DNA analysis now gives these descriptions a scientific grounding. In addition, the study of Justinian-era Yersinia pestis strains allows comparison with later pandemics, revealing evolutionary patterns that may inform how pathogens emerge and adapt over centuries. While the Justinian Plague eventually subsided, possibly due to changing climate conditions, declining populations, or herd immunity, its legacy influenced urban planning, trade networks, and even political stability in the Byzantine Empire. Modern scientists also emphasize that understanding historical pandemics is crucial in anticipating potential future threats, as it underscores the interplay between microorganisms and human societies across time. Although the research is robust, some questions remain about the precise routes of transmission and the role of other co-infecting pathogens, inviting ongoing investigation and debate. In conclusion, the recovery and analysis of ancient DNA have provided a remarkable window into the Justinian Plague, solving long-standing mysteries about its origin, virulence, and impact, and offering perspectives that resonate with both historians and epidemiologists today. And that's something worth remembering when we next consider how interconnected human health, environment, and history truly are.